I've always been fascinated by the use of mathematics to understand complex biological systems. Years ago, my team and I worked with a scientist who was using mathematical models to analyze disease networks as dynamic systems. What struck me was not that his models contained a complete description of the biology. Of course they didn't. Part of the network were missing, and some of the interaction were still poorly understood. But that was precisely what made the approach useful. When we combined the mathematical model with experimental data, the discrepancies showed us where our understanding was incomplete. The model did not magically fill the missing biology. It helped us identify where to look for it and which experiment would be useful to reveal it. More recently, I have become fascinated by the idea of modeling entire cells and eventually tissue, organs, and maybe in a not so distant future organism. I have written several articles about why we might want to do this, how it could work, and what it could make possible. One objection comes up again and again. How can we model a biological system that we do not fully understand? My answer is that is exactly why we build models. A scientific model is not a claim that we know everything. It is a mathematical description of what we think we know at a particular moment. And we use it to make predictions, confront those predictions with experiments, and learn from where it fails. A good model is not necessarily complete. Sometimes its greatest value is that it is wrong in an informative way. Physicists have worked like this for generations, so I see no reason why biology should be different. That is why I'm always looking for papers that take the modeling of cells seriously. And this new paper in Cell is definitely one of them. So it is asking pharmaceutical companies for 500 clinical trials, and preferably the failed ones. As a thing, we tend to publish things that works, but when we train model, we also need the thing that didn't work, and this is typically the type of data that you can never get your hands on. They want the patient-level clinical data, molecular profiles before and during treatment, and samples that might explain why one patient responded and another did not. The authors argue that without this kind of data, AI will not become genuinely predictive in cell biology or medicine. It may classify cells, it may reconstruct familiar patterns, it may produce a very impressive benchmark, but it will still struggle with the question pharma actually cares about. What will happen when we give this treatment to this patient? That is the real argument in this paper, and it is a much more interesting argument than the title suggests. First, this is a perspective, not an experimental study. There is no new model, no new dataset, and no benchmark result. 15 authors are trying to set an agenda for the field. Their inspiration is interesting because it brings us back to the beginning of this video, mathematics. So they talk about David Hilbert's famous list of 23 mathematical problems from 1900. The biology version contains 15 challenges from molecular interactions all the way to clinical trial outcomes. And the affiliation of those authors is interesting. Several authors are connected to BioHub and the Chan Zuckerberg Initiative. So the corresponding author, Andrea Califano, leads BioHub New York. Shana Keeley leads BioHub Chicago. And BioHub plans to reach 10,000 GPU by 2028. And its head of science, Alex Reeves, started and led Meta ESM protein language model project. So this is not an anti-AI paper written from the sideline by any means. It is a warning from inside one of the largest bets on AI-powered biology. Scaling the current approach may not be enough. The paper starts from a simple observation. AI has produced remarkable results in protein structure. I know that for a fact. My background in structural biology, so for me AlphaFold was definitely a career-altering moment, let's put it like that. It worked also in protein design and sequence-based biology, but those wins do not automatically transfer to cell, tissue, or patient. A protein sequence can be represented as an ordered string. A cell cannot. Cell behavior emerges from regulatory networks, metabolism, epigenetics, spatial context, and other cells. Many effects are not pairwise, they depend on combinations. The authors use the assembly of a 60S ribosomal subunit as an illustration. It requires the coordinated binding of up to 47 proteins. If you consider possible combination of that size across the enormous repertoire of regulatory motifs and protein isoforms, the theoretical search space reaches roughly 3.48 times 10 to the power of 150. That is larger than the estimated number of atoms in the observable universe. This number is a warning, not a calculation of how much training data a model literally needs. Biology does not search every possibility uniformly. Physical constraint and evolution eliminate most of the space. That is also the author's solution. Do not ask a generic transformer to rediscover biology. Build molecular networks and other biological priors into the models. The second problem is data. Language models trained on trillions of tokens. The paper puts the largest cell biology dataset around 10 to the 10th or 10 to the 11th biological measurements, far less and scattered across assays, tissues, and experimental conditions. So the shortage is not just about volume, it is about comparability and purpose. A billion steady-state cell profile will not necessarily teach a model how a CD4 T cell responds to a new cytokine combination. Then the paper gets more, let's say, uncomfortable. In 2025, Allman, Elze, Huber, and Anders compared 5 single-cell foundation models and 2 other deep learning models with deliberately simple baseline. The task was to predict transcriptomics changes after genetic perturbation. None of the deep models beat the simple baseline. For double perturbation, adding the 2 individual effects was often as good as or better than the sophisticated alternatives. The cell offer suggests that this failure may be structural rather than incidental. Or in plain English, perhaps the answer is not simply a larger model and another year of training. They also challenge the benchmarks. Cell type classification is useful for methods development, but it is not biological discovery. A statistical significant improvement can still be useless at the bench. Their most provocative example is the precision recall curve. They argue that if a biologist can only afford to test predictions at a false discovery rate below 5%, most of the curve being reported is irrelevant to the actual experiment. So I agree with the principle, although 5% is not a universal law. The actionable threshold depends on the cost and risk of the experiment. The larger point survives, measure the part of the model performance that changes a scientific decision. Their proposed fix is sensible. Tier 1 is retrospective methods development. Tier 2 is prospective biological discovery with predictions locked before the experiment begins. The 15 challenges are organized into 4 layers. At the molecular interaction level, regulatory and signaling interaction, epigenetic interaction, and cell-cell communication. At the molecular function level, synthetic mechanisms, going from genome sequence to biochemical function, and drug mechanism of actions. At the cellular and system level, genome to phenotype, cell state reprogramming, synthetic circuit design, and systems level mechanisms. Then come the 5 translational challenges: biomarkers, toxicity, efficacy, organismal responses, and clinical trials outcomes. Calling these problems is slightly generous. Many are research programs, large enough to occupy several institutes. The authors acknowledge this and add experiment, success metrics, data requirement, and non-generative baselines. So this is one of the paper's best choices. A generative model does not get bonus points merely for being generative. It still has to beat linear regression, lasso, random forest, network models, organoids, CRISPR screens, or whatever the relevant alternative happen to be. Challenge 11 is biomarker discovery. The proposed bar is demanding: a clinical AUC of at least 0.9 in a blinded prospective cohort, a panel of 20 features or fewer, and validation in at least 2 independent cohorts across demographic groups. Challenge 12 is toxicity. The paper uses the TGN 1412 trial, in which all healthy volunteers developed a cytokine storm despite reassuring preclinical results. Their benchmark is prediction of grade 3 or worse adverse events with at least 80% sensitivity at 90% specificity. Challenge 13 is efficacy. The point is that a good drug can look ineffective in the wrong population. Trastuzumab is the obvious example. Without selection for HER2-positive tumors, its effect will have been diluted across an unselected breast cancer trial. There is a wording problem in the paper here. It says about 15% of patients carry mutation that sensitize their tumors to trastuzumab. The 15% is defensible. Modern estimates often put HER2-positive disease around 15 to 20%, but mutation is not the usual mechanism or clinical definition. Trastuzumab selection is based mainly on HER2 protein overexpression or HERB2 gene amplification. The main argument remains right. Patient selection can separate a failed trial from a foundational medicine. Challenge 14 is organismal response. The example is predicting vaccine response from a person's pre-vaccination immune profile. Prediction must be sealed before dosing. The model should achieve an AUC above 0.8 for distinguishing high and low responders, and a correlation above 0.7 between predicted and measured antibody titers and T-cell responses. Most importantly, it must beat existing approaches such as linear regression, lasso, and random forest. Challenge 15 is the big one. Predict responder and non-responder in prospective phase 2 and phase 3 trials. Reach an AUC of at least 0.8 and validate the proposed sensitivity or resistance mechanism in patient-derived organoids or CRISPR screens. The last requirement matters. A prediction is more useful when it tells you what to test next, not merely who sits on which side of a curve. This is where the paper moves from scientific critique to an industrial proposal. The authors say the greatest barrier is insufficient high-quality training data. Public trial records usually reveal outcomes and response rates, not the molecular profiles needed to explain those outcomes. Some relevant data are locked inside companies, some are sitting in biobanks, and some never existed because the right samples were never collected in the first place. Their answer is a public-private consortium model on initiatives that have already pooled pre-competitive data. The target is at least 500 trials in 5 years with matched molecular measurement at baseline and during treatment. Start with failed trials where commercial sensitivity may be lower. Use CDISC standard for clinical data and community standards such as GA4GH for multiomics. Put access, publication, and model evaluation under independent governance. Use a honest broker such as DREAM to run blinded assessment. Company will need a reason to participate. The paper suggests early access to consortium models, co-authorship on benchmark publication, and credit for contributions that support FDA-recognized biomarker qualification. I think it's a serious proposal, but 500 trials is a mobilization target, not a magical sample size calculation. Incompatible trial across unrelated disease, assays and endpoints do not become one training set. The value will come from consistent sampling, usable consent, provenance, longitudinal measurement, and harmonized endpoints. So the hard part is not counting trial, it's making them interoperable without stripping away the context that gives the data meaning. So I will put 3 questions back to the authors. First, are these really generative AI challenges? Biomarker classification, toxicity prediction, and responder stratification are predictive tasks. They may eventually benefit from generative model, but classical machine learning, causal models, and system biology may solve part of them first. The paper partly recognizes this through its baseline requirement. I will go further. The goal should be predictive biology, not victory of a particular AI model family. Second, how will biological priors be kept from becoming biological prejudices or bias? Interaction networks are incomplete, and published biology is biased toward popular genes and well-funded population. A model constrained by yesterday's knowledge can reproduce yesterday's blind spot. The authors mention dynamic reassessment of priors that need to be central, in my opinion. Third, who owns what comes out of a consortium? Andrea Califano and Mingxuan Zhang disclose a patent filing on Gremlin, which puts molecular interaction network into the attention mechanism, but does not invalidate the paper, and it is only one possible architecture. But if pharma contributes the data, model access, intellectual property, publication rights, and benefits sharing must be negotiated first. And yes, the authors use Claude Sonnet 4.6 to check references, redundancy, and grammar. Honestly, that is not hypocrisis. It may be the most perfectly aligned AI used in the paper, a narrow task, human accountability, and explicit disclosure. So for years, pharma has asked whether AI can reduce the failure rate of drug development. This paper turns the question around. Have we created the data, experiment, and prospective benchmark that will allow any model to prove that it can? Right now, the answer is mostly no. The 500 trial proposal is therefore worth pursuing, but pharma should not participate as donor handing over failed study to an AI project. It should participate as a co-architect, defining the scientific question, the data standard, the validation rules, and the commercial terms. Because failed trials are not waste. They are incredibly valuable, and I think right now we are not using them enough. And handed property, they may be the most valuable training data and training material the industry has never learned from.